I will quickly go through the last week lessons. In last week, we talked about the hierarchy of organisms, which is start from atom to biosphere, atom, molecule, micromolecule, cell, tissue, organ, then organ system, organism, population, community, ecosystem, and biosphere. Those are known as the hierarchy of organisms. Then we talked about cells and tissues. What are cells and what are tissues? Then different type of tissues and different type of cells and how different type of tissues are going to form. Those things we discussed in the last week. And then we move to the cell theory. There are three pillars of cell theory. We talked about them. Then we move to the main type of cells. Main type of cells. Then we categorize the cells into prokaryotic cells and eukaryotic cells. I told you this is the important part. Prokaryotic cells and eukaryotic cells. Prokaryotic mean I told you this is the kind of cells which not origin actually, which emerge in the world, for the first time. Prokaryote mean before nucleus. That means this prokaryotic cell doesn't has any nucleus. Okay. Actually, the significant difference between prokaryotic cells and eukaryotic cells is eukaryotic cells has a nucleus, but prokaryotic cells doesn't has a nucleus. That is the main significant difference, and there are a lot of differences. And when you move to the kind of differences, then there are no any membrane-bound organelles or membrane-bound nucleus in prokaryotic cells, but in eukaryotic cells, you can see membrane-bound organelle. The only organelle in the prokaryotic cells or bacterium cell is ribosome. That is the only organelle which we can see because ribosome doesn't has a membrane. Okay. Then we talked about the structure of a prokaryotic cell. Then we talked about reproduction examples, and we moved to the eukaryotic cells, and we finished from the last part, reproduction. Okay. Today we have to start from types of eukaryotic cells. The types of eukaryotic cells. Can you tell me what are the kingdoms comes under to eukaryotics? What are the kingdoms? Last week I told you. What are the kingdoms? And what are the kingdoms comes under to prokaryotic cells? Can you remember?
Kingdom domain,
Sorry.
kingdom fungi, kingdom bacteria. Sorry,
Uh,
domain
domain mean? Kingdom domain mean? What comes under to domain? Actually, there are three domains. No? There are three domains. One is bacteria, other one is archaea, other one is eukarya. Eukarya. These are the domains, okay? Then bacteria domain and archaea domain comes under to prokaryotic cells. So otherwise, we can see prokaryotic cells in bacteria and archaea domains. Then when we come to eukarya, there are kingdoms. There are kingdoms. Then being the this hierarchy is known as domain, no? This is the domain. Hierarchy is domain. Then after domain we can see kingdoms. Kingdoms. Okay. Are you clear?
Yes, sir. Clear.
Then kingdom Plantae, kingdom Animalia, kingdom Fungi, kingdom Protista likewise. Those are known as the kingdoms. And those kingdoms comes under the eukarya domain. And in that kingdom, you can see eukaryotic cells. That means if you take a plant cell that comes under to kingdom Plantae, that is the kind of example for eukaryotic cell. Then if you take a animal cell that comes under to kingdom Animalia, then kingdom Animalia is a kingdom of eukaryotic domain. That's also a kind of eukaryotic cell. Fungi, same. Protista, same. Understood?
Yes, sir.
Okay. Before we move into the okay, let's move to the plant cell. Let's move to the plant cell. In a plant cell, you can write this comes under to kingdom Plantae. Put a hyphen and write. Oh no, hyphen. Write in within bracket. Kingdom Plantae. Kingdom Plantae. Kingdom Plantae. Can you remember the classification under comes to kingdom Plantae? Under kingdom Plantae, how plants are going to be classified? What are the two main groups? What are the two main groups? Can you remember? In this lesson, in this same lesson, we have talked about Plantae. Can you remember? This can be classified into two main groups. One is flowering plant, another one is flowering plant, another one is non-flowering plant. Non-flowering plant. Then flowering plant again can be categorized into two groups as what is that? Monocotyledonous plant and dicotyledonous plant. Okay. We have to learn about this classification and we have to learn about the differences between monocots and dicots. Okay. In our next lessons. Okay. Then plant cell. The plant cell is made up of mainly from cellulose. Okay, that means cell wall is made up of cellulose. Cell wall means the outermost outermost layer. Cell wall is the outermost layer. Outermost layer. Outermost layer. Okay, sorry about that one. Then this outermost layer is known as the cell wall. You can see this yellow color region is the cell wall. Okay, yellow color region is the cell wall. Actually, it works as a wall. Works as a wall. That means it can protect the entire cell and it gives a shape to the cell. What it is does, it gives a shape to the cell and the cell is protected by this cell wall. Understood? Do you understand?
Yes, sir.
Okay. Then plant cells have a large central vacuole. A large central vacuole. This is a significant characteristic in a plant cell. That means if you are given to identify a plant cell and an animal cell, okay, a plant cell and animal cell, we can identify the plant cell by this central vacuole. Okay, the animal cell doesn't has a vacuole. Only the plant cells has vacuole. Sometimes animal cells can have a temporarily small vacuole, not but there is no any permanent vacuole. Okay. Then next one is chloroplast for photosynthesis. Chloroplast for photosynthesis. This is the chloroplast. This is the chloroplast. The process of photosynthesis is happen within this chloroplast. The process of photosynthesis happen within this chloroplast. And again, this is a kind of thing which you can only see in a plant cell. Okay, only seen in a plant cell. In the fungi cell, you can see any chloroplast. In animal cell, you can see any chloroplast. Okay. Then there are no centrioles. This is not very important for you. But this cell wall, okay, when we talk about the cell wall, animal cell doesn't have a cell wall. Animal cell doesn't have a cell wall. That means you can use this tree. You can use this tree characteristic to distinguish between plant cell and a animal cell. To identify a plant cell and animal cell, you can use cell wall, vacuole, and chloroplast. Understood?
Yes, sir.
Okay. We have a comparison in this tute later on, so you can easily memorize that things. Okay, just keep in mind this 3 4 point. That's in. Then in an animal cell, in here there is no any cell wall. Okay, there is no any cell wall in here. There is no any cell wall, and there are no vacuoles. There are no vacuoles. And there is something called centrioles, those are present, and there are lysomes for digestion. Lysomes for digestions. Okay. Then next one is fungal cell. This is a fungal cell. This is a fungal cell. And in here there are no chloroplast, but cell wall made up of chitin. This is important, okay? Cell wall is made up of chitin and this can have multiple nuclei. This one can have multiple nuclei. Multiple nuclei. Clear?
Yes, sir. Clear.
Okay. Then can you tell me What are the things which can commonly seen within this among these cells, plant cell, animal cells, and fungal cells? What are the common things? What are the common things?
So Golgi complex.
Sorry, I didn't get
Golgi
you.
complex.
Golgi complex. Okay, okay, that is a common thing. What else?
Mitochondrions.
Yes, mitochondria are there because mitochondria are the power station. Those are the power station to work, or to happen the metabolic activities. Energy is needed, no? So that energy is provided by the mitochondria. As a common thing, you can see cytoplasm. Okay, cytoplasm mean the this region. In here, the yellow color region is the cytoplasm. Here, green color region is the cytoplasm, and here is light brown region is the cytoplasm. Okay. The importance of the cytoplasm is all the organelles are embedded in this cytoplasm. Can you see? All the organelles are embedded in this cytoplasm. Embedded in this cytoplasm. And any living cell has a plasma membrane. Any living cell has a plasma membrane. Plasma membrane is just like a outer cover which we can see in between cytoplasm and cell wall. Okay, cytoplasm and cell wall. It just like a wrapping paper. Just like a wrapping paper. Cytoplasm. The cytoplasm are plasma membrane. Okay. These are the common things. Okay. Common things. And we can see DNA and nucleus in here. In every eukaryotic cell, we can see DNA and nucleus. And ribosome, not ribosomes, chromosomes also there. DNA, nucleus, and chromosomes also there. Okay. So I think it is better to, or it is easier to remember the differences in between cells, no? Because there are a lot of same or common things. There are a lot of same or common things. So I think it is better to remember the differences. Differences. We have to do some MCQs today. So I will give you a discussion for every, not every MCQ. I will provide 3, 4 discussions. So at home you can write the discussions for every MCQ. Then I will tell you how to write that one. Then we talk about some of the some of the organelles, no? So we have to talk about these organelles in the next lesson.
Sir,
Yes,
sir, is it important to note down the function of each organelle?
each organelle. No,
Yes,
it
sir.
is in here, no?
Oh, okay, sir.
It is in here. Here also there. Okay, then let's move to the main structural differences between prokaryotic cells and eukaryotic cells. Okay. This is the structural differences between prokaryotic and eukaryotic cells. Okay. First one is the nucleus. Nucleus. Highlight this point. Okay. Nucleus. Then there is no true nucleus. There is no true nucleus, but we can see genetic materials are freely available in the cytoplasm. But we can see genetic materials are freely available in the cytoplasm. freely available in cytoplasm. But in eukaryotic cell, there is a membrane-bound nucleus, and within that nucleus, we can find the genetic material. Within that nucleus, we can find the genetic material. Okay. In these prokaryotic cells, there is something called plasmid. Plasmid. This is also a kind of structure which has genetic material, that means DNA, and then there is something called circular DNA. Circular DNA. This circular DNA is used for mainly for genetic engineering purposes. Genetic engineering purposes. In here, right, plasmid and circular DNA also available. Plasmid and circular DNA also available under prokaryotic cells. Plasmid and circular DNA also available.
Done, sir.
Okay. Uh, Two membrane bound nucleus. Okay, that's enough about that one. Then membrane bound organelles. We can see in prokaryotic cell there is no any membrane bound organelles. Okay, membrane bound organelles. Here it has given us except ribosome. That mean that doesn't mean this ribosome has a membrane. Okay, this ribosome has a membrane. It doesn't mean this ribosome has a membrane. This ribosome also doesn't have a membrane. Understood?
Yes, sir.
I think cut this word except, otherwise it will mislead you. Cut the word except. Okay. The next column, present, yes, mitochondria ER. What is mean by ER? What is mean by ER? Have you heard about this one? ER? ER mean endoplasmic reticulum. Endoplasmic reticulum. There are two kind of ER. One is SCR, that means smooth endoplasmic reticulum. There is another one RER, rough endoplasmic reticulum. Okay, if this ER has ribosomes, okay, if this ER has ribosomes, it become RER. That means rough endoplasmic reticulum. No need to write that one, okay? Because we have to talk about in the next table. But in the last week, I told you there are some organelles which has dual membrane. That means there is a two membrane organelle, double membrane organelle. Okay? Can you remember what are those? I told you there are three organelles which has dual
Chloroplast,
membrane. Yes, chloroplast. What else?
nucleus,
Yes, chloroplast, nucleus. There is another one.
mitochondria.
Mitochondria. Yes, mitochondria. Mitochondria. Can you write down this one? And write double membrane. Double membrane. This is important. Double membrane. Double membrane. Finish writing.
That's it.
Okay. Genetic material organization. And here also I give you there is a circular DNA and plasmid. And both of these cells has chromosomes. Okay. Both of these cells has chromosome, but in prokaryotic cell there is only single circular chromosome. Single circular chromosomes and there are no histone protein. Histone means a kind of protein, okay? A kind of protein. There are no histone proteins. That means chromosomes are usually like this, no? This shape. How many chromosomes do we have? How many chromosomes?
46.
Yes, there are 46 chromosomes within us. And that means we have 23 pair of chromosomes. 23 pair of chromosomes. I drew two chromosomes and by this pink color, I have indicated the histone proteins. Histone proteins. And this kind of structure we cannot see in prokaryotic cell, but in eukaryotic cell, we can see multiple linear chromosome associated with histone protein. Associated with histone protein. Actually, this histone protein, this point, this not asked for A level yet, okay? Not for asked for A level yet. That means I'm not sure in this year will that be checked or not. I'm sure that will be checked very soon. So you have to remember that one, especially you have to remember that one. And I think it's better to draw this figure under eukaryotic cell, that means here. And name these are the histone proteins. The small figure is a small figure. Most of the points in this table has been asked for A-level SFT paper. But this histone protein stories, not yet.
That's it.
Okay. Next one is cell size. Usually, usually, usually prokaryotic cells are smaller compared to the eukaryotic cells. Prokaryotic cells are smaller compared to the eukaryotic cells. Okay, that is the point we want to know. Prokaryotic cells are generally smaller and eukaryotic cells are generally larger compared to prokaryotic. And at this point, to observe prokaryotic cell, you will definitely need a microscope. Definitely need a microscope. But some eukaryotic cells, to observe some eukaryotic cells, you don't need a microscope. Okay. Only some, not everyone, not every cell, but only some cells are quite large, quite large. Then let's write in here. Need a microscope for observing. Need a microscope for observing. Oh, folks, need a microscope, folks.
Done, sir.
Okay. Sasi, are you writing?
Yes, sir.
Okay. Then ribosomes. Ribosomes. I told you we can see ribosomes in both type of cells. We can see ribosomes in both types of cells because ribosome doesn't has a membrane. Okay, ribosome doesn't has a membrane, and so we can see ribosome in the both types of cells. But there is a small structural difference. There is a small structural difference. That means prokaryotic cells have 70S ribosome, but eukaryotic cells has 80S ribosomes. 80S ribosomes. Okay, 80S ribosomes. But there is a special case. Mitochondria and chloroplast has 70S ribosomes. I don't think this point is checked for SFT paper, but remember, just as remember or just as a memory about this one. I don't think this point will be checked, but there is something like this. The only